Welcome to the RedChip Virtual Conference.
Thank you for attending the Galectin Therapeutics presentation. My name is Jim Czirr, as previously mentioned, Executive Chairman, and I will be certainly looking forward to answering any questions when we're through with this presentation. You've all seen forward-looking statements and understand what those mean. Investment highlights. Galectin Therapeutics, we have proprietary compounds, which are first-in-class, that actually inhibit galectin proteins, which are very instrumental in advancing a number of serious diseases. Our compounds are all carbohydrates, which give them a very, very favorable safety program. We have two compounds in development, GMCT01, which is used in cancer. Another one, GRMD02, is used in liver disease, like non-alcoholic steatohepatitis, as well as cirrhosis. The science is validated. We have preclinical models that show that galectins are critical targets for the intended diseases that we're treating. We have very large market opportunities. You look at diseases like NASH, and you're talking about markets that could be as much as 15 million people with no alternative therapy at the moment. Intellectual property has all been developed in-house. We have sole ownership. There's no license arrangements, which, as you know, gives us a much greater valuation than someone who's had the in-license and is giving away 20% of the future royalty that they could make. We have an experienced management team, and speaking of the management team, I want to focus on Dr. Peter Traber, our CEO and CMO. Peter's got over 25 years experience in the biomedical business. Probably for this conversation, it's probably best to focus on the fact that at one time he was the Chief Medical Officer in Glaxo in charge of global drug development. He's very excited about the technology we have here, and that's why he's willing to work for a small biotech company for a fraction of his salary. Ansel Kleosis and Ellie Zomer are very, very experienced people in carbohydrate chemistry and both production as well as drug manipulation and drug development. And everyone else is well suited for their job. If you look at the key milestones over the next 18 months, for starters, we have a symposium that we're hosting. This is the second symposium, and we are producing a book on galectins. There's only one book on galectins on the market today, and we are responsible for putting it together. But we're hosting this international symposium that's going to be attended by people from all over the world that are leaders in research in the area of galectins. In cancer, we have started injecting patients on our stage 1 and stage 2 phase 2 trial on metastatic melanoma. Testing our compound, which is an entirely different paradigm in cancer treatment. We'll talk about that in a minute. In the first quarter of 2013, we expect to have top-line results on the first 12 patients. In fibrosis, we are looking to achieve a qualification with the FDA for injecting people by the end of the year. We expect to initiate our phase 1 trial sometime in January or February. We expect to have phase 1 data by October and start our phase 2 trial by the end of the year. Science of galectins. Let's take a look at galectin proteins and see what they're all about. They're actually critical in the development of many fibrotic and neoplastic diseases. Normally, cells will secrete these galectin proteins in small quantities, and they are typically involved in cell-to-cell matrix interactions, promote cell-to-cell growth, and act like a cellular glue. But we find that they're markedly increased in the diseases of inflammation, fibrosis, and cancer, and that galectin-3 is the most prominent one of these proteins. So our molecules are novel complex carbohydrates that actually bind up these galectins. If you look at the left-hand side of your screen, you'll see these long polymers with multiple binding sites. They bind up the galectins and inhibit, what they do. The binding is very strong on galectin-3, and we disrupt what it would normally be doing. Again, we have a very, very low toxicity profile because these are carbohydrates, and as you know from your high school chemistry, carbohydrates break down into CO2 and water. We have two classes of compounds, a GR and a GM compound, and we've got very low manufacturing costs because these are made out of seeds that are available by the shipload. On the intellectual property, we have our GMCP class, We have composition of matter patents that have priority date of 2003 were issued in 2011 and should be able to get a few more years put on them because of that. We have 5 U.S. patents. We have 13 patents granted internationally with 5 pending. We have methods of use in liver fibrosis as well as methods of use in NASH. On our GMCR series of products, those are a little bit more recent developments, and they have priority dates of 2011 and 2006, and those are still pending. All intellectual property is generated in-house, no encumbrances. And we have, and right now it's interesting to note there's no established generic pathway for being able to duplicate a complex carbohydrate drug. So even if patents run out, there'll be some proprietary knowledge, and anyone who wants to compete is going to have to go through the entire clinical trial development process. Let's look at immune enhancement in cancer therapy. Research indicates that these galectins are involved in a variety of activities on behalf of the cancer. They promote angiogenesis, the growth of new blood vessels. They are involved in detachment and adhesion, metastasis, migration, and also in tumor immunology. We're going to focus on tumor immunology and the galectin effect that protects tumors from the immune system. That's right. That's what I said. There's a galectin effect that protects tumors from the immune system. And this is a cartoon caricature of what's been determined and discovered by Dr. Pierre van der Bruggen at the Ludwig Institute of how tumors use galectin-3s to defeat a patient's immune system. You can see here in the cartoon that galectin-3 is being excreted by the tumor, and it is coating the surface of the T cells that would normally be secreting cytokines and killing cancer cells with those cytokines. And you can see that it stops that process and arrests that from happening. This is a major, major problem with every cancer patient. It's believed that virtually every type of tumor excretes galectin-3, and it's surmised that this is happening in all those situations. What we found is that when we introduce our molecule, GMCT01, that it blocks the galectin-3 and it inhibits it from binding to the T cells and allows them to continue to create cytokines and kill cancer cells. Research indicates that we not only can prohibit this, but that we actually can reverse it once it's happened. And so this is a slide we're looking at here is a nice sigmoid curve that shows that as we increase the exposure of cancer cells to, I should say T cells to the GMCT01 molecule, that you see a marked increase in the amount of gamma interferon produced, which is the chemical they use to kill cancer cells. This slide that we're looking at here shows the regenerative ability to restore the ability of human CD8 T cells to kill tumor cells. You're looking at the bottom at, well, let me explain the experiment. There were cancer cells and their analogous T cells taken from 3 different patients from the fluid around their tumors, and then separated into different concentrations. So you can look at the very left-hand lower corner. You see 0.01. That's a 1 to 100 ratio of T cells to cancer cells. And as you go across the bottom, you see that the ratio of T cells to cancer cells has increased for each of the 3 different sets of T cells and cancer cells from the 3 different patients. You can go all the way over to the line between 1 and 110, which is a 5 to 1 ratio. So we've gone from a 1 to 100 ratio to a 5 to 1 ratio, a 500-fold increase in concentration. And you see from the chart that there's very negligible amount of change in the percentage of tumor cells killed during the 20-hour experiment. When you repeat the experiment and you add the GMCT01 molecule that we have, you'll see a marked increase where you can get as high as 45% of the cancer cells killed during that same period of time, indicating that our molecule can reverse this galectin effect that has defeated T cells. So we've also already demonstrated safety of the GMCT01 molecule by having it in over 100 patients. We had a phase 1 trial with 40 subjects and saw very strong safety data. We conducted a couple of phase 2 trials. The company at the time didn't have money to complete them and had different management. But the results were that in a colorectal cancer metastatic model with end-stage patients that were line 3 and 4, we saw that we actually, by adding our molecule in combination with 5-FU chemotherapy agent, we saw that we improved median survival roughly 46% from 4.6 months to 6.7 months. And as you can see, a comparative would be that Erbitux had a similar number of 6.1 months. We also noticed that there was a severe decrease in the amount of severe adverse events. We only had one case of grade 3 or 4 mucositis, no cases of grade 3 or 4 diarrhea. And for those of you in the cancer space, you would know that diarrhea would normally be 12 to 40% of the patients would have it, and 17 to 24% of the patients would have mucositis at the levels of 5-FU we administered. So, we're seeking partners to develop the drug in combination with chemotherapies. And as I mentioned, the safety data was extremely strong. So what are we doing with it now? Well, with this understanding of this galectin effect and how tumors use these galectin proteins to turn off the patient's immune system's effectiveness, we have entered into an arrangement with the Ludwig Institute, who's conducting a clinical trial using a vaccine that they have been developing. They have used this in a number of clinical trials. They've had very nice immune system responses and increase in T-cell count, but they have not seen any significant improvement or any measurable improvement on median survival. That's what drove them to do research to actually determine and discover that there was a galectin effect where tumors were using galectins to turn off the immune system. So this is a proof of concept study. It's a phase 2 study that's got 2 stages, stage 1 and 2. In the first stage, there's going to be 12 patients in 2 different sets of patients that are going to receive a vaccine every 21 days to build the T-cell count. And then they're going to receive GMCTO1 every 3 days, and then while they continue to get the vaccine every 21 days, in order to reverse this galectin effect and or protect from it occurring, and the hoped for result of improvement. The timeline on this is that we've already dosed the first patients, and there's an ongoing recruitment process and classification. We expect top-line data by the first quarter, the January area. And if we like what's going on, we will continue. We could continue with the stage 2 portion. We could, stop it and go into a phase 2B controlled trial for FDA approval. We unquestionably would like to start looking at seeking partners at this time, because this would be the proof of concept that galectins can be inhibited in this process that they use to defeat the immune system can be reversed. This is potentially applicable to all types of cancer, and this would be a major discovery, and we think would be the basis for entering into a joint venture arrangement with maybe more than one company. So immune enhancement by blocking galectin effect is potentially synergistic with many types of emerging cancer therapies. We've used it with chemotherapy. Well, if you think about it, what type of therapy wouldn't be better for cancer if you had the ability to keep the patient's immune system turned on and protect it from the galectin effect? So all of these new vaccines that are being worked on, and there's 2 agents that have been approved, Provenge with Dendreon and Yervoy with Bristol Myers Squibb. But there's
Welcome to GoToMeeting, online meetings made easy. There are 1 other callers on the call.
tumors, and that those T cells will be subject to the galectin effect if they're not protected. And so we think we've got a very strong enabling technology here. The vaccine market is growing significantly, but this would not be limited to just the vaccine market. We think we're best positioned to advance tumor immunology with galectin inhibitors because of our experience. Now, there are some competitors in the space, We know that Galectin Biotech in Sweden is a discovery stage company that's working on small molecules. We haven't heard of any of them coming anywhere close to a clinic at this point in time. Ohio Pharmaceuticals purchased GCS 100 from Solana, and this is a product that's been in a sequence of bankruptcies with a sequence of companies. Mandel Med has been working on a truncated galectin-3 protein, but hasn't done anything with it for 5 or 6 years. Didn't have much results, and the Stellic Institute is working on an RNAi approach to inflammatory bowel disease, which of course is not relevant to what we're doing. So we think we're best positioned to advance this project into humans and into an approval. So let's change gears for a moment and go look at our second platform, which is liver fibrosis. Liver fibrosis is a very serious problem. Cirrhosis of the liver, claims 40 to 60,000 lives a year. It's hard to get real specific and current data. Some of this data is a little bit old, but nonetheless, we know it's a serious problem. We know there's no treatment for it. That when people have cirrhosis, that their only treatment is a transplant. And as you can see, there's a very small number of transplants available each year for the large number of cirrhosis patients. We know that NASH is contributing, becoming a very large contributor to the number of people that are developing cirrhosis. We know galectin-3 is a critical target for the therapy of liver fibrosis. And how do we know that? Well, we know that galectin-3 is produced in large amounts by fibrotic liver tissue, whether it's animal tissue or whether it's human tissue. We know that galectin-3 is essential. In different animal experiments, they have actually done experiments that were with animals that were deficient that had the galectin-3 gene knocked out. And without the galectin-3 gene, it was impossible to get scar tissue or fibrotic growth in the animal's liver. Very strong marker. So we also know that the human stellate cells that are responsible for growing fibrotic condition in humans, that when we block galectin-3, we turn off the production of the collagen, which scar tissue is made of. So, and then in laboratory animals, we've been able to actually reverse the fibrotic condition. If you look at this next slide, we're looking here at the results of injecting thioacetamide into a set of rats for 8 weeks. You can see all the red scar tissue that is rippling through the liver tissue and shouldn't be there, and is responsible for the disease cirrhosis. Now, this has long been thought to be an irreversible problem and situation. And when we take half the animals and treat them with our drug, we end up with livers that look like this on the right, almost totally clear of any underlying scar tissue. This is the slide that was responsible for getting Dr. Traver to join the company and take the CEO position. We've done some studies with mouse models, and we've been able to watch this disease develop. And it starts out with fatty liver disease, develops in the NASH in 7 weeks, and you get fibrosis in the advanced stages of NASH, and ultimately you get the cirrhosis conditions. We ran an experiment to see whether or not this would not only reverse scar tissue and potentially a treatment for cirrhosis patients, but what it would do for fatty liver patients, NASH, non-alcoholic steatohepatitis. So there are roughly 9 to 15 million people in the United States that have this disease, according to the NIH. And of course, the United States does not have a corner on this market. It's a result of bad diet and type 2 diabetes. And you can see from the vehicle slide, all the white dots, those are animals that are diabetic, that have been fed high-fat diets, and just like your neighbors that eat incorrectly, they've gotten all this little fat tissue throughout the liver, as well as inflammation and ballooning hepatocytes. The score on the right is the pathologist collectively scoring both the vehicle and the slides where the animals were treated with RGR and VO2 product. And you can see the scoring with the blue dots on the slide that the animals treated with GRM VO2 saw a significant reduction in the combination of fat, inflammation, and deteriorating cells. But people don't die from a little fat in their liver. They die from the collagen or scar tissue caused by the inflammation that is a result of the fat. And you can see here in this next slide that there's a set of animals that were treated during the development of the disease or early treatment, and another set that was treated after the disease was more established or late treatment. In both cases, you can see that the diseased animals in black had elevated levels of collagen. And collagen or scar tissue is what kills people. If you look at the animals that were treated with the GRMBO2 illustrated in blue, you can see a significant drop, and that drop goes down to normal. As you can see in the center between the two graphs, the normal level of collagen in a mouse is 0.33, and they have been returned to normal. This is a very, very significant finding because there are, like I mentioned, there's up to 15 million Americans with fatty liver disease, and there is no pharmaceutical treatment available. Just to complete the circle here, we've gone ahead and stained the slides for Galectin, the presence of Galectin-3, the culprit that causes the scar tissue or initiates the scar tissue. And you can see that the untreated animal has a lot of Galectin-3 stained in brown, and the animal treated, the liver is treated with GRMBO2, have a very minimal amount of Galectin-3 left. So this slide takes a little more time than we have today, but I want to just show you this in general. We're talking about the process here of how extracellular matrix or collagen gets laid down. There's a number of metabolic syndromes that can start the process of creating the cytokines that trigger this. What I want to show here is that Galectin-3 is involved in all of these. If you look at the hepatocytes, bile duct cells, the macrophages, if you look at the quiescent stellate cells that when they become inflamed start producing the cellular matrix and the collagen, Galectin-3 is in all the pathways. Now this is flies in the face of traditional thought about having a very specific drug that binds to a very specific target that's in only one spot on a disease, and we think this is one of the reasons why no one has found a treatment for liver disease and fibrosis previously, is because they were focusing on affecting the hepatocytes, or they were focusing on the macrophages, or they were focusing on the stellate cells and some receptor that was involved with those particular cells. Galectin-3 is involved in all of them. And if you've ever played college football or watched American football, you know that if you want to keep your quarterback from being sacked, you have to stop, all the different people that are rushing and trying to tackle the quarterback. In this case, we believe that you need to interfere with all the different pathways that are contributing to the inflammation that is triggering the growth of the scar tissue. And we believe that we do that. So let's look at our timeline. We're very excited about this. We are in preclinical at the moment, which are going very, very well. Primates have had, huge doses of our molecule, and many, many, many times the therapeutic dose range without any side effects. We're very optimistic as we head into our FDA meetings, and that we'll be able to get our first patients enrolled in January, February, and that we'll be able to complete the phase 1 trial, by Q3. We then expect to start the phase 2 trials as quickly after that as possible and finish it in 2014. We expect at this point in time, with good results, that we will get numerous offers by large pharma and that we will cease to exist because the market is so big and there's no competition. We'll obviously be testing with biopsies and looking at other serum markers to illustrate success. I don't know if anybody's followed the story with Inhibitex or Pharmacet. They were two companies that were recently bought at the end of phase 1 for $2.5 billion, and another one at $11 billion at phase 2. That had a hepatitis C drug. While hepatitis C is different than NASH, it's interesting to note that both of them end with scarring and fibrotic liver damage and ultimately death. One of them, the hepatitis C, is 4 to 5 million patients in the United States with drugs that benefit about half the patients. There's 9 to 15 million NASH patients with no treatment options at all. Examples of therapeutic approaches, there have been a lot of things tried. Bottom line, nothing has shown any significant results at all in being able to make a difference and affect the disease. So we believe that GRMDO2 is well positioned with respect to competition. We believe we have the most attractive mechanism with multiple sites of action in the disease and a very well established target. We also noticed that our treatment is independent of hyperglycemia or hyperlipidemia, so we don't have to stop the animal from being a diabetic in order to fix the liver problem. This may reverse established fibrosis. It certainly has in animals. And again, we have a very low toxicity profile. Many of these other approaches and compounds have actually failed because of liver damage, which is an interesting irony. If we look at Milestones, again, we're going to be hosting an international conference. This is a chance for a significant amount of publicity and exposure. We have a lot of collaborations with independent scientists around the world. In the cancer, we're expecting, we've already infused the first patient here, and we're expecting top-line data. This top-line data, this is an uncontrolled trial, so it is possible to report data as it develops. And so if there was a significant percentage of the initial patients actually saw their tumors shrink substantially and perhaps even a cure, we have something here that could find itself into the mainstream media and be a major topic of discussion well before year end. We have, but the top line data nonetheless would be handled by somewhere in the first quarter of 2013. In fibrosis, we are expecting that we'll have our IND by year end, and which is only a few months away. We believe we'll initiate our phase 1 trial by February and have results by October, and that we will get our phase 2 trial started shortly thereafter. Finances and capitalization, there's 15.6 million shares outstanding. You can see there's a variety of preferred and warrants. There's this thing called the 10X fund that I organized when we came back in and resurrected the company in 2009. A number of those shares, including the Series B warrants and the Series B stock on a converted basis, are all locked up till 2015. You can see the exercise prices. It's significant to know that management and officers and directors own a substantial part of this, and the public float is not that large, and a very small number of non-affiliates. Investment highlights, again, proprietary compounds, first in class at binding and inhibiting galectin proteins. These are complex carbohydrate drugs with highly favorable safety profiles. The GMCT01 enhances tumor immunology, and MDO2 is a potential treatment for non-alcoholic steatohepatitis. The science is validated. The mechanisms of action of both of these compounds are well characterized and understood. When you have a molecule like galectin-3 that is necessary for the creation of a disease like fibrosis, you can't get a better target. We mentioned large market opportunities. This is something that you contemplate and think about is significant, because large being a value statement doesn't tell you how large. But when you're looking at a drug that could treat 15 million people that have a disease that is going to rob them of quality of life and ultimately become cirrhosis and take their life, it's a very, very significant type of need, and it's a huge patient population. I don't know of any other patient population that has 15 million people in the United States that don't have a drug treatment. So you're looking at drugs that could be as large as any drug has ever been, and those are very desirable by our larger partners. Intellectual property, again, we have invented all of this in-house ourselves and do not have to license it and not licensed in anything. To put this in perspective, if you were to pay a 3% royalty on an in-licensed patent and then turned around and licensed it to Big Pharma for 15% royalty, you our valuation would be 20% higher because we don't have to give up the 3%, which represents 20% of the ultimate, relicense fee. Very experienced management team and experienced board. We haven't had a chance to talk about it previously, but we have a scientific community that has 7 of the key opinion leaders in the world of liver research that have signed on to develop our NASH clinical trials and be part of our clinical trial development team. We have 4 of the world's leading experts in NASH that have already signed on to be part of the development team and to help conduct clinical trials. Dr. Scott Friedman at Mount Sinai Hospital in New York, one of the giants in liver research, is an actual consultant to the company in addition to being on that clinical trial development team. So with that in mind, I want to thank you for attending, and I'll take questions and answers.
Jim, thank you. First off, I just want to say thank you for taking the time to present today. We do appreciate it, and we will jump right into Q&A.
Okay.
First, I want to clarify, just to make sure, there's been a couple questions here. Do I understand correctly that your products, GM-CT01 and GR-MDO2, are both products made from non-toxic carbohydrates, and what kind of impact does that have on success rates?
Well, it's pretty common understanding in the drug development world that over 50% of all compounds that enter the clinic wash out because of some unintended liver metabolite or toxic side effect. We've got experience with 100 patients in our cancer drug and have had 0 severe adverse events of any significance. And so it's a very strong additive. I mean, you could argue that it has twice the opportunity of success of when you're first entering a phase 1 trial, because you've eliminated 50% of the reasons why drugs fail. So when you do an analysis of the risk-adjusted net present value, it has a much higher probability of success.
Thank you, Jim. Next question here. You touched base on this, but could you give a little more color into the clinical trials in phase 2, and what would be the earliest you would see results or indication of success or effect in that trial?
Well, that's an interesting question to ponder. The trial is set up, and we expect to have top-line data sometime in January, February. And this is kind of a rose-colored glasses approach, and so I'm a little reluctant to answer a question like that. But because of the way the trial is designed, we are in a position to be able to release data prior to the accumulation of all top-line data. Because we're in a disease like metastatic melanoma, which is a severe disease that takes down virtually everyone that it infects, if we were to have the first 3 patients, for example, have tumors shrink 50%, and or let's say one of them be cured, that would be something we could announce, and that could happen as early as end of September, 1st of October, which is only, again, 4 or 5 months away. And that's the kind of thing that it would be newsworthy. The New York Times type of newsworthy.
That's great. That's exciting news there. Upon the results or potential results from a phase 2, is that when we would see interest from Big Pharma on a buyout? I know you touched base on that briefly. Could you maybe give a little more color into that as well?
Yes. Phase 2 is when you have your first real signs of efficacy, where your trial is designed to capture efficacy data, even though we are going to design our phase 1 trial for hints at efficacy. We believe, because of the size of the market, that we're going to find large drug companies excited about talking to us. And again, because it's so big, we think that the Pharmaset and Inhibitex examples, indicate what Big Pharma is willing to do when they have an opportunity to have a very significant drug compound. So we're expecting to get offers to be purchased at that time.
Thank you. And that market size, I believe you said, was around 15 million. Is that correct?
Well, there's estimated to be 15 million patients. In all fairness, there's a continuum of degree of disease. You know, as people get diagnosed with, or have, I should say, not diagnosed, but they have fatty liver disease or non-alcoholic steatohepatitis. And the initial stages, a lot of them might not even know they have it. But as the disease progresses, it becomes more and more of a deterrent to their health and their lifestyle, and ultimately, enough inflammation is created that there's enough galectin production, and the scar tissue gets triggered, and now where the cascade has started that is going to lead to death. So on one end of that 15 million people, you've got people that are in severe need that may already be cirrhosis patients, waiting for a liver transplant. And on the other end, you have people who, have started the process but are, 15 years away from having it becoming a critical situation in their life. So, but we've run some numbers, and if you took just 1 million patients out of the 15 million and charged $300 a week for treatment, which is a nominal price for a treatment of a disease that would take somebody's life, and I'll put a 25% discount rate in it for time value of money, and run the entire gambit, you come up with risk-adjusted net present value numbers that are well in excess of $1 billion. So it's a very big market. I mean, if you look at those numbers again and said, well, if you charge $10,000 a year per patient, and you had 1 million patients, that's $10 billion. That's almost as big as Lipitor.
Excellent. Thank you for that. Lastly here, do you have a location or a venue for this international conference you're planning on hosting in September?
Yeah, it's going to be in Boston at the Harvard Club. A number of our scientists are, ex-Harvard professors, and we're using their access.
We'll wrap up Q&A now. Jim, I appreciate your time again for taking the time to present at the RedChip Small Cap Virtual Conference.
Well, thank you very much. It's been a pleasure.
That was Jim Czirr, Executive Chairman of Galectin Therapeutics, trading under the symbol GALT on the Nasdaq.
This video is not a recommendation or solicitation to purchase or sell any security, nor does it constitute investment advice. Visit RedChip.com for full disclosures on RedChip clients.